Thank you guys all for coming. I'm so excited to introduce Dr. Fred Gould. He was my advisor for my PhD. I actually looked up stuff to tell about him because I had to have good stuff to tell. So he's at NC State in the Entomology and Genetics Department. He's the William Neal Reynolds Distinguished Professor of Entomology. He's high up there. His main focuses are in ecology and evolutionary biology. And he started out his work at State as a postdoc. He started there as a postdoc, and throughout his research, he's always looked at ecology and evolution, but some of his work was really groundbreaking in the Bt crops and looking at insects and how to get them to prevent them from becoming resistant to Bt. So a lot of his work was really helped with guidelines and regulations for the EPA on how to grow anything Bt. All of his work was involved with that. And he's currently doing more research on genetic engineering, looking at all different pest species and how to use genetic engineering to help control these different pest species. I gotta give him some credit for some of his awards. So in 2000, he's got too many awards to actually go through and name. But some of them, so 2011 he got the Holiday Medal for Excellence, which is the highest honor for an NC State faculty. He also, in 2011, was inducted into the National Academy of Science. I also want to mention that's the year I was finishing my PhD, which might be coincidence of why he got those. And then in 2013, he was awarded the UNC System Oliver Max Gardner Award, which is the highest award for a faculty in the UNC system, so throughout all North Carolina schools. And that's usually given to people who've made one of the greatest contributions to the welfare of humanity. He's really up there. His newer work, so he's working with all kinds of different pests, and he started a program at State for graduate students in interdisciplinary program looking at genetic engineering for pests. And he's also the co-director of the Genetic Engineering and Society Center at NC State, which is a fairly new, couple years old, right? So he's going to be telling us about genetic engineering in the 21st century and how the current technologies and how that's going to impact us as a society.
All right, great.
Welcome.
Okay. So, let me see, I have to get out of this and go back. So, the real title of this is Living in a Genetically Engineered World. And it's great for me to come and talk to younger people about this because we are starting to live in a genetically engineered world. But by the time you're my age, the question is, what is this going to look like? What does this world look like? And I guess what I want to do in this talk is give you a little preview of what it might look like. But actually, what it looks like will depend upon you and other people your age in terms of making decisions about how society accepts or doesn't accept technologies and where it all goes. So that's what I want to talk to you about. And Rachel started out talking about Bt, so I'm assuming maybe people know about this. But how many of you, know, I think you all hear the word GMO all the time. You all know that, right? So we were very interested in this when it started because in North Carolina, if you try to grow sweet corn, this is what it looks like if you didn't use insecticide spraying. And typically, it would take 11 sprays of insecticide every year in North Carolina to get corn to look like this. But this is sweet corn that never was sprayed, but it has this Bt gene in it. So basically, there was an organic insecticide that was from a bacteria, Bacillus thuringiensis, and they took a gene from that that codes for a toxic protein, and it's toxic only to caterpillars, and they moved it into the corn, right? And right now, if you go to Walmart, you'll get Bt corn. There was a big controversy over Walmart taking this on, but most of the field corn in the United States and most of the soybeans are genetically engineered for one thing or another. So I guess I could start by asking, do you guys know what it is that you eat that's genetically engineered? What kinds of plants?
Soy.
Soy, what?
Orange.
Oranges, no. So let's go down the rundown. What could be genetically engineered? What? Wheat? No.
Chicken?
No. Right. Soybeans? Yes. Corn? Yes. The cotton in my jeans? Yes. Rice has been engineered, but nobody's growing it commercially, and we'll talk about that a little bit. So we'll talk. I want to talk about what it is that's happening, what's engineered, what's not. But this is just sort of a start, right? Is genetically engineering plants, and not everybody likes that, right? I think you probably have heard people protesting this. Does anybody here have a friend or a relative who won't eat genetically engineered? Some of you.
They think they're not eating.
Ah, they think they're not eating it, right. And maybe even some of you would rather not be genetically engineered. How many of you think that genetically engineered foods may not be safe? Some of you. Yeah. Actually, if you ask the general population, more than 80% think that genetically engineered foods might not be safe. So it's an issue. So there's a real issue actually in Mexico about genetically engineered corn. And they're the ones who brought us corn, right? So sometimes it's a question about safety, about sort of scientific things, and sometimes it's about culture. And Rachel was talking about a group we have, and I'll talk about them later, of graduate students. Well, one group of them is very interested in agricultural pests, and their first trip was down to Mexico, and this is at the oldest agricultural university in Mexico, outside of Mexico City. And I don't know if many of you have heard of an artist named Diego Rivera, very famous Mexican artist. And he did all the murals in a chapel at that school. And this is one of his paintings. And most of his paintings have corn or maize in them. But this was a special one that struck me. Because remember, all these different kinds of corn, every village you go to has its own kind of corn, and they're good for specific things. And they will not use American corn to make tortillas. It just doesn't taste right to them, right? So they all have special corns, and they all have special properties. But what's interesting about Diego Rivera's painting here, you can see the corn, but if you look at the roots of the corn, there are two dead people here. These are the martyrs of the Mexican Revolution. So you can see how ingrained in their culture maize is. They see those plants coming out of the ground being based on their whole culture, right? So these are their heroes. I mean, think about, we put George Washington underneath, maybe a cherry tree or something, whatever it is, right? We don't do that, right? This is very important to them. So when all of a sudden what they brought into the world is now owned by companies, and those companies are trying to sell it back to them it's like, and you hear this, you're raping our corn. We bring this, and you do all your crap with it, and then you give it back to us, right? And you compete because the corn that comes from the U.S. is much cheaper to produce than this. So they're both scientific issues about safety and ecology, but they're also these cultural issues. So just to ask you folks a question, if you go to a fancy restaurant in Raleigh, sometimes you'll get salsa, and it'll be in this special Mexican bowl or something like that. Sometimes in a hotsy-totsy restaurant, that's going to be pretty expensive. And how do you get all these nice colors? If you go to the supermarket, you'll see peppers. When I was a kid, this was a pepper, right? Now you get all of these different colors, and it's very nice. Makes your. So if you go to a fancy restaurant, they may tell you about where those peppers came from. Well, they went into the Andes Mountains and they found these wild peppers, and they bred those colors into the peppers that you were eating in your salsa. And the idea is that somebody going to that restaurant will just feel better thinking that there's this connection between them and the Andes and whatever, right? Do any of you ever think about that? Where your food's coming from? Does it come from the farm or where it comes from? So if you went to that same fancy restaurant and they told you that those peppers actually came from here, because we genetically engineered them, how would you feel about that? Would you pay extra to go to that restaurant? Maybe not. So what is it about that? Why are you more comfortable, or do you feel more connected to something when it comes from nature than when it comes from a laboratory? You know, so there's these things that go on in terms of your world. So this is, you know, maybe the natural world, and this is the beginning, potentially, of a genetically engineered world. So if you go to the tropics, sometimes you'll see these beautiful butterflies, right? I mean, that really stand out. And once in a while, you'll see some of these actually in the temperate zone around Raleigh, right? But most of our butterflies are kind of boring. So what if we could fix that butterfly and make it fancier, so that when you walked out in Raleigh, you'd have fancier butterflies to look at? What's the difference between the appeal of finding something natural that's beautiful in the woods versus us engineering something, I don't know, maybe a little gaudier or whatever, but that's there, right? So what's this all about? So what about human beings? Spider-Man. How many people have seen the movie Spider-Man? Good. All right. So how did Spider-Man get his powers?
Bit by a spider?
Bit by a spider.
Radioactive spider.
What's that?
Radioactive spider.
No, genetically.
What?
The
You're
original.
too old. Wait a
I'm
minute.
too old, yeah, sorry.
So what was that spider?
It was genetically engineered in a lab.
Genetically engineered in the lab. Yeah, and if you remember, I have a little clip of that movie, but that's the spider fighting him. It almost looks like a black widow. But in the movie, it's genetically engineered. But, hey, I'm old too. That was Spider-Man, right? So back in the day, Spider-Man got his powers from being bit by a spider, but this is the way it happened. Accidentally absorbing a fantastic amount of radioactivity, the dying insect, and it's not an insect, but it's okay, in sudden shock bites the nearest living thing at that split second before life ebbs from its radioactive body. Right? That's what it was. And then Uncle Ben, Spider-Man's dad or uncle, right? With great power, there must also come great responsibility. You know, Spider-Man, was kind of full of himself in the beginning and all of that, right? So what's interesting with the comic books was that in the year 2000, Ultimate Spider-Man came out in comics. And from 2000 to 2009, there were comic books, but the spider became genetically engineered. So even in the, so curious, right? So what's happened here? So when I was younger, we used to watch all of these movies. There must be 100 movies. You're laughing. Some of you know these, right? They're great old movies. The one I love the most was The Incredible Shrinking Man, right? So they go through this cloud of radioactivity, and all of a sudden this guy starts shrinking to the point that he's smaller than his cat, and he gets smaller and smaller because he was exposed to radioactivity. This was sort of like this thing that was going on when I was growing up. People were, both scared of the Cold War, the power of radioactivity, the unknown. What could it do? What's the chance that radioactivity is going to make you, you've taken biology, Think about all those organs in that. What's going on? But today, how many of you have seen this? Come on, how many of you haven't seen that? Right. Jurassic World. This is not Jurassic Park, right? Right? In Jurassic Park, right, you bring back dinosaurs from the DNA that you find. But then things sort of drop off because the dinosaurs aren't so exciting anymore, right? So they have to engineer a better dinosaur. And that's what this is, right? So what has happened here in terms of our imagination, somehow or other? I mean, you could have had the switch to anything at all, but this switch from radioactivity, this big unknown nuclear energy, could be wonderful, could be terrible. All of a sudden, it's replaced by genetic engineering. I mean, we had plenty of Godzillas and all sorts of monsters that came out of radioactivity. So what's going on here? this is a award-winning book called The Wind-Up Girl. And it's about this future where the world is taken over by calorie companies, basically, that make genetically engineered food. And they have all the patents and all of this, and it's this whole big thing. But it takes place in Thailand, mostly. And what they figured out is how to genetically engineer animals as well. So they actually engineered these cats and other things that have become problematic for them. But they also engineered wind-up girls. And wind-up girls look like anyone else, except they walk a little bit different, so you could tell if it's not a real human being. They specifically put that in there so that you could tell. And in Japan, the wind-up girls are revered as being better than human, lacking our foibles. In Thailand, they're hunted down, not like animals, because animals have a spirit. They don't have a spirit because they're not real. They're genetically engineered. So what defines what matters, right? I don't know how many of you saw a movie called Her, which is about this guy who has this relationship, right, with a computer, who also has a relationship with 6,850 other people, right? But what about, and so you pull the plug on that, right, and it's gone, but it wasn't real, or was it real? And what about a wind-up girl? What makes something matter and what doesn't? So when I start talking to you about a genetically engineered world, where will we be when you're my age? All right, so, but what do we really have here? All right, so we definitely have corn and such. We don't have, those peppers come from the Andes, that's fine, right? But how long, right? And how long before we decide that we'd like a better butterfly?
We already got one in our pigs, so.
Yeah, but we don't have these genetically engineered butterflies, but we certainly have genetically engineered mosquitoes. Okay, and this is a special mosquito that I'm fond of. It was developed in Colorado, and it's Aedes aegypti. It's a mosquito that transmits dengue fever. But this one has been engineered so that it produces an interference RNA in its midgut. Okay, and this is a female, and the green, the glowing green, is really just a marker to tell you the gene is active, right? But it's producing an interference RNA that is focused on interfering with the development of the dengue virus. So this is a female, and this gene only gets turned on in the female after she's taken a blood meal. Right, so think about that, right? When you take a blood meal from an infected person, you bring the virus into the insect, it multiplies in the insect, then when it bites the second person, it transmits it. But if you can shut it down, its machinery, then it can't multiply in the insect. And that's exactly what that insect's about. Right? And this is early days for these kind of things, but a lot of people are working on this for malaria. All right? But when this first, I want to bring you back, because we're pretty far along now, so I'm talking about genetically engineered world. When this first idea happened was back in 1970s. And this is 1975, when the first people who started to engineer bacteria and phage, right, viruses, realized what they were able to do all of a sudden. I mean, now that's, we know this. But they called a meeting in a place called Asilomar out in California, where they all got together and they decided that they better put a moratorium on doing this work because they weren't sure about what kinds of problems there would be from that. And two of those people sitting around the table were Paul Berg and David Baltimore, and they wrote an article with a statement about how to be careful with this new technology that they had. And, one of the things that they wrote was nothing is known about the potential infectivity in higher organisms of phages or bacteria containing segments of eukaryotic DNA, and very little about the infectivity of DNA molecules themselves. Right? So they're starting to mess around with this. They know very little about it. And they, as scientists, decided we better put a halt to this until we know more. So this happened, and then they figured out more about it. But that was the 1970s. So lots happened since that meeting, and these folks both, they won Nobel Prizes, etc. So this is Obama in front of a genetically engineered cornfield, right? Yeah, these are pigs that have been genetically engineered to be environmentally friendly. They don't produce as much phosphorus that contaminates waterways. And then, of course, those pigs have never been commercialized. Don't worry, you're not eating them, or if you would love to eat them, you can't eat them. But these are genetically engineered fish called glowfish, and you can buy them at PetSmart or Petco, unless you live in California where they're illegal. Right? So a lot has changed from this time, and these two folks have also changed. This is what they look like now. Right? And what's interesting is that last March, they wrote a new article. And the new article is a prudent path forward for genomic engineering and germline gene modification. Last March, something really major happened. And that is that people got to the point where they've moved from engineering these bacteria with bacterial viruses to the point that they can engineer human beings. And it was a new technology called CRISPR, and I'll get into that. All right? But all of a sudden, this was possible, and Chinese scientists actually published a manuscript where they had engineered embryos that couldn't go to gestation, they couldn't become people, but they showed that they could engineer them. So all of a sudden, in the same way that they had that thing with the bacteria, in '75, these old guys said, whoa, so here's David Baltimore, Paul Berg, you know, they're now a little bit older than they were, coming up with this article saying, hey, let's have a meeting about this. But one thing about this paper is it wasn't just the old guy. There were some women involved here. And one of the most important ones is Jennifer Doudna, who's really a big hero or such for some people in this story. And this is Jennifer, and she is the one who actually wanted to pull this meeting together. She is in a battle with some folks. She's at Berkeley, and she's in a battle with some folks at MIT about who's going to have the patent on this whole thing. Worth billions of dollars, according to people, right? So it's really complicated in terms of what's going on and who's meeting and everything now. So I just want to say they're having a meeting that they actually, a number of national academies, National Academy of Sciences in the United States, National Academy of Sciences in China, and others are putting on December 1st through 3rd about International Human Gene Editing Summit. And if anybody's interested, you can go to this URL and sign up to be part of that meeting. So they're trying to open it up. So they're also having a lot of social scientists there. It's a little different atmosphere than it was when they were having that Asilomar conference. This is an article you could find, I think it's 2 weeks ago Sunday in the New York Times Magazine section called CRISPR Quandary. And it's really about Jennifer Doudna, what she's been up to. And you can see what it's talking about, right? Who are we now that we can be engineered? So I just want to tell you a little bit about what this is. Okay? So what they discovered, and there's somebody at NC State who was a major player in this, looking at fungi, they found that fungi have this funny way of defending themselves against invading viruses. Right? Same way we have an immune system. They have this weird immune system where when a DNA virus comes in, they take a snippet of that DNA and they make it part of their genome. And they also have a gene that produces a protein that's called an endonuclease. It cuts DNA. And what the fungi have done naturally, because they don't need us, they have figured out a way to come up with this, what's called a Cas9 protein, and these guide RNAs, right? They take the DNA and make the RNA. The RNA gets together with this Cas9. And remember, they took the DNA from this infected virus, put it into their own thing. So the next time they get infected by that same virus, they use this RNA to find the DNA of the virus, clomp onto that, and cut the virus with this endonuclease. And it takes both of those things. But what Jennifer Doudna and other people figured out is, gee, well, it's just a protein. We can code for that, right? We just take borrow that from the fungus, And then we can make our favorite RNA to look for places in the genome we want to cut and replace with something else. And so they came up with what's called CRISPR. And I'm not going to go through what that acronym is. It's about a palindromic chain of these things that comes to make that. But just to understand, basically what it does is it cuts DNA. So, what are we going to use that for? Are we going to use that to cure diseases? Are we going to use that to make everybody taller? What are we going to do, right? What is going to happen? And what's going to happen depends on you folks in the end, right? Depends on the public interacting with researchers. Or is this technology going to get so easy that people could do this without government approval? So it's a whole different world here that may be coming. But what was really interesting to me was we all talk about the human side, of course, as an entomologist, even though it's a spider, it's not an insect, but it's very interesting to be thinking about what's going on non-human side. What do these CRISPRs mean for us as entomologists? And so, you know, do you ever think of how could a genetically engineered insect biting somebody make them into Spider-Man? Oh, I don't know. But at the same time, last spring, when that paper came out about genetically engineering humans, another paper came out, and this was in Nature, I guess, or Science. Anyway, Science Express. It was in Science. And it's called the Mutagenic Chain Reaction, a method for converting heterozygous to homozygous mutations. So that was kind of a weird name for things. Mutagenic chain reaction. And it scared people, and it, I guess rightfully so. So the deal is you all have been learning about, right, Mendelian inheritance, right? So if we assume that you only have one chromosome, right? So this is a nucleus, and inside is your diploid, and you're a heterozygote for some mutation. And you mate with somebody else, and they don't have that mutation, right? You all know that the expectation is if these are the sperm, half the sperm get this, and if this was the case, it was in the male, right? No matter what, the offspring will look like that. Half are going to be heterozygous, half won't have it. You start out with 1 out of 4 copies having this mutation. You end up with 1 out of 4 copies having this mutation. What this mutagenic chain reaction of turning heterozygotes into homozygotes means that you start out as a heterozygote, but by the time you produce your sperm, they both have the mutation. So basically, the DNA gets cut by this CRISPR, and it uses the other template. We get chromosomes broken all the time, and humans have a way, and so do all animals, of fixing that. And the way they fix it is by moving this into that slot to repair the DNA. So you start out being a heterozygote, you become a homozygote, and then you pass that on to both of your sperm. So instead of 1/2 being heterozygotes, all your offspring are heterozygotes. So you started out with a frequency of 1 in 4 of the DNA molecules. Now you wind up with 1/2. So if you think about that, usually you'd expect, they did this experiment with Drosophila, and instead of half of the offspring turning out to have this mutation, about 94% of them did. And that's their experiment showing, right? So you have this ability to start out at a low frequency, put this into a population of insects, and have it take over. So one of the problems about that mosquito that I told you about, right, was you could breed it in your lab, make it, and it wouldn't transmit dengue. Well, that sounds great. You've made this mosquito that can't transmit dengue, but then, okay, so you release it, and so you have your mosquito that can't transmit dengue, but if there are 1,000 other mosquitoes that can, what have you accomplished? But if you had this mutagenic chain reaction on your side, you could get this as a heterozygote to be homozygous, and over time, not too much time, you turn the whole population into ones that can't transmit. And people are really working on it. So far, the Gates Foundation has put in about $100 million into this technology, and they're continuing, and they're getting ready to move some of the mosquitoes to Africa. So there's a big thing going on, and I have to be on their ethics committee. So they're very concerned about how they do this, right? They don't want to be another Monsanto. They want to be very careful about how they're doing it. But it could be, one of the real problems with this is that if you release this, because the Kenyan government would really like to get rid of malaria using this genetic technique, but the Tanzanian government is opposed to it on ethical grounds, how do you stop those mosquitoes from crossing borders? So there's this trans-border issue that's really important that they're dealing with. So, right now, we're very interested in mosquitoes that could transmit dengue fever, and the same kinds of things hold. So what's interesting is it's easier right now to create a mosquito that has this ability to just move. What we're trying to do is create a system where it's sort of localized, that it can't move beyond a certain border, or that it only works for 10 years and then goes away. But there's a lot of things in here. But one of the things to bring up is what happens if you let these mosquitoes out and they look like they're working great. They decrease malaria, right, which is a big issue. Save millions of lives, like 500,000 children die every year from malaria. So it seems like a great thing. Somebody will get a Nobel Prize, right? But what about if that works for 10 years? And what happens then is so few people have had malaria that what's called herd immunity, right, because you build up immunity if you get malaria, has gone down. And if the parasite finds a way around it, and then it comes back with a vengeance. So you have to really think through what you're doing in this genetically engineered world. So with this whole thing about the CRISPR, people were working on this before this new technology came out. There were other ways of doing it, but it took a lot more funding, and that's why most of that $100 million from the Gates Foundation was spent before CRISPR technology came out. There were other technologies, I'm not going to talk about them, but there were others. But when this came out, there was this, all of a sudden, this big ideas about what you could do with engineering non-human organisms. And this is an article that was in a new journal called eLife, and they were talking about this RNA-guided gene drives, right? So remember, there's the protein, but it's the RNA that guides it to a specific spot. And so the idea was you could use this for threatened and endangered species, to save them. You could use this to eliminate rats from the environment, especially on islands and stuff. We'll talk about it. Agriculture, all of these different things you could do with that. So the question is, if you can, should you? So one of the things we're very interested in is, about Darwin and the Darwin finches, right? So one little bird gets over to the Galapagos Islands. There's all these different habitats. It evolves to use those. Well, the same thing happens on a lot of islands, and this is the Hawaiian Islands. And the Hawaiian Islands has a different bird that got there, and it became the Hawaiian honey creepers. And you can see how different they are. Well, right now, so also, Hawaii didn't have mosquitoes. What a place, huh? But now it does. And it also has invasive birds from the mainland that get over there. Well, birds from the mainland have been dealing with bird malaria forever. And so they've developed resistance to it. But the Hawaiian honey creepers didn't have bird malaria, and most of them are very susceptible to it. So when this little mosquito got over there, and it transmits bird malaria, it's one of the things that's causing these to become endangered. So they're being pushed up the mountainsides in Hawaii to places where these mosquitoes aren't and where there isn't as much competition from the birds from the mainland. But as people think that as global climate change happens, this is going to get smaller and smaller. Right, so there's a real problem with this. So the idea is, can you genetically engineer this mosquito so it can no longer transmit bird malaria? And another thing is, in terms of rodents, most of the extinctions on islands of these birds and other things are actually caused by rodents that either eat young chicks or that kill plants. And actually this group that we're working with, Island Conservation, tries to get rid of these rats and mice from islands. And the way they do it is they come in with big helicopters and they drop rat poison all over the islands. And there are people who protest against this. It's a complicated kind of thing. Sometimes it works, sometimes it doesn't. But in the Galapagos Islands, finally, after 100 years, this tortoise, this Galapagos tortoise, had its first babies that have survived. If that tortoise didn't live so long, it would have been extinct. So now that they've gotten rid of the rats that eat the baby tortoises, now they can survive. But there are problems. This is the tsetse fly, so it transmits trypanosomiasis. You may know of it as sleeping sickness, but it's worse in cows, so some cows get this and die, and it's hard to do agriculture when you don't have oxen to plow and you don't have milk. So a lot of people have been trying to get rid of the tsetse fly. And potentially you could do this with genetic engineering. But one thing to recognize here is that this area where tsetse fly exists is also the area on the Earth that's the highest in biodiversity. So if you get rid of tsetse fly, it could be that this would really be good about alleviating poverty in some parts of Africa, so you have more development, right? But you also have to recognize that you lose some biodiversity, and that might be a good trade-off, right? Lower poverty, but less insects, whatever, right? But the question is, if you actually did this, if you did it wrong, you'd probably have more poverty, right? Because of inequality. You know, when you find oil in a certain place, right, doesn't mean people are going to be less poor. So there are a lot of social questions that are going to be facing us as we move forward. So when we started out at NC State, and we were working on genetically engineered corn, and we started working on these mosquitoes, we realized what was going on with Monsanto and everything else, but we were a bunch of biologists, ecologists, molecular biologists, population geneticists. Rachel did actually incredible work on this mosquito in Mexico, partially while she was pregnant. She was down there. And she did these great studies that nobody else has ever done anything like it in understanding their ecology, to know more about how this could be done well. I should have thrown in a slide of you doing this. But this is really...
all seen me.
What's that?
I show pictures
Oh,
of it, so probably all seen me.
okay. But this is really important to put so we call this interdisciplinary. And it was hard for everybody to talk together. But what we recognized that biologists couldn't do this alone. And we moved to this thing of bringing in social scientists and ethics people on campus to work with us, and formed an even bigger interdisciplinary group that really had trouble talking to each other. And we had faculty and students from all these different areas, from the molecular genetics to people who work on rhetoric, to understand how we convince each other of things, how we talk to each other. And political science and history was very important in this group. And we wrote a grant to the National Science Foundation to fund us to do a project on genetic engineering in society, the case of transgenic pests, those things that I've just been talking about. And after 3 tries, we got funded. And this was a big $3 million grant, and we brought in graduate students. So this was only for graduate students. But I want to tell you about this because I don't know where you're going to go in your life, but you could think about these kinds of things. And the idea was, most of the time when you go to graduate school, you go in molecular biology or ecology or something like that, some people talk about getting a PhD is learning more and more about less and less until you know everything about nothing, right? Well, that's okay. But the idea here in this program was that instead of just that, we bring in students who had undergraduate degrees in these different areas, bring them together to work on one project. And that project, I'll tell you, so that would focus them on one thing. And so the first group was working on these mosquitoes in Iquitos, Peru. And you see all the tarps here because it rains a lot. This is the largest city in the world that you can't reach by roads. It's right on the Amazon. And they went down there and did work, and our first graduate just got a faculty job. The second group was working on those mice. And they went to islands off the coast of California, one inhabited and one uninhabited. That's a sort of a place where they've gotten rid of some of the goats and pigs on those islands that also were causing problems, and looking at that. And the third group was the one that went to Mexico, to Oaxaca. I showed you that picture of the corn and the people, that's where they were. And so this was the agriculture there that they were studying, trying to understand, could there be an approach to getting rid of some of their storage grain pests using these transgenic pests. But anyway, and we'd have, so this was our group, and Some of the students we brought with us were Mexican students also, so there'd be interactions. So just to say, I mean, these are things that are possible for careers. Is to today, as opposed to years ago when you'd just be in one narrow slot, there's a bigger push, and industry and government are looking for people who have these broader kind of perspectives. And so we've gotten a big group at NC State now doing this kind of thing, and that's what Rachel said. It's a Genetic Engineering Society Center, and we were able to hire some new people in the social sciences, from humanities, political science, and from economics to help us to look at these kinds of questions. So that's what we're up to, and what we're interested in, talking about this genetically engineered world, we focus on issues that have to do with genetically engineered crops, and one of those is golden rice. How many people have ever heard of golden rice? Oh, quite a few. Okay, so golden rice is a rice that's been genetically engineered to have genes for, in the beginning, from daffodils, but now from corn and stuff, that increase the beta-carotene in the rice. So one of the problems is that over, it's like 500,000, 250,000 people go blind every year from vitamin A deficiency. And in a year after that, half of them die. So this is a big issue. It's as big as malaria in a way. And the idea is if you could engineer this rice to have higher beta-carotene, that could help with these problems. It's a very big political issue, but it's something that's coming along. Other issues that are coming along are these genetically engineered fish that grow twice as fast as regular salmon. They've been ready to be made commercial for 15 years, and the FDA has never given them the authority to do that. They grow them now down in Panama. Another is reviving extinct species. You may know there are people working on the woolly mammoth and the Carolina parakeet and other things. And then there's genetic engineering of humans. So these are things that this group looks at. Okay? But these are things that everybody as a citizen has to look at. So I guess I just want to leave it in that thing to sort of just have maybe a little bit of discussion, if we have some time, to get your perspectives on that. So I'll leave it at that. Thank you.
So golden rice has been around for, what, like 2 decades? What is the main issue surrounding it if it provides vitamin A to a rice-based culture?
Yeah. So this is a complicated issue. So, you know, it's been a decade since the first genetically engineered rice was created that had slightly higher beta-carotene. And who's the guy who wrote the book on slow foods? Anyway, there's been a lot, Michael Pollan. Anyway, people who've been very critical of it, saying that, gee, a kid would have to eat 5 bowls of this stuff to get enough, even half enough of that beta-carotene. And the thing is, he was right a decade ago. So you hear on one side people saying, oh, you know, a decade ago we could have had genetically engineered rice, saved all these lives, but it was those anti-biotech people who kept it down. And then you have people saying, oh, it's all phony, there's nothing to this. It never would have worked. And then there are the people who are actually doing the work. And so they've come along from this using daffodils genes to increase this, to using other approaches. And now those levels of beta-carotene that are in rice now are quite a bit higher and could actually, I think, just with one helping of rice be about half as much as you need per day. So it's gotten better. But the International Rice Research Institute that's doing all this work in the Philippines, if you go to their website, they're very cautious about that. You know, they're just trying to do their work. And they know that if they had a variety of this stuff that was really looking good, some people went off and tested it in an unethical way, which is a problem. But they've been testing it in Bangladesh and the Philippines and other places, and the first thing they had, the yield wasn't right. Some tested did well and some didn't. You're not going to get a farmer to grow this stuff if they can't get good yield. So they went back to the drawing boards. They now have a different variety. And if you go to their website, the International Rice Research Institute, you'll see what they're doing. That's a good question, because basically, you know, when it comes down to it, you know, there's all this fluff, and then there's the people who are really doing the work and dealing with the issues. They are trying to get things done right, to test this. One of the complaints is if somebody is in poverty and is malnourished, they're not going to be able to transfer that beta carotene to become vitamin A. And so some studies have been done. So it's not just genetically engineered foods that could have higher beta carotene. People have been using conventional breeding to increase the beta carotene in sweet potatoes and corn. So there's a corn now that's really orange, and people have tested that in Africa and found that people who are vitamin A deficient can increase their vitamin A content by using it. Yes?
As a follow-up, so the whole, I know there's different kinds of rice, but are you going to run into the same issue as in Mexico where you have different types of varieties of corn? Or is this going to be accepted by rice farmers in rural China?
Good question. So one of the things you'll see on their website is they're back-crossing these genes into the most important cultivars of different countries. I used to work here as a consultant, and the Thai people would say, I wouldn't feed this Philippine rice to my dog. You know, I mean, it's that same kind of thing as with the maize, right? It's very culturally important. So a question is, who's going to feed yellow rice to their kids, right? So, well, maybe in India where saffron rice is a big deal. Or maybe if your kid is going blind, right? But it used to be this kind of thing that the USDA used to give out this butter that would be, extra butter, and they'd always mark it from the USDA. Nobody wanted to be seen eating that butter because it meant you were poor. So it could turn out that this is why there's a lot of social issues here. And it's sort of like, it's how you do it. How do you introduce this, right? So I think you're asking really good questions here. Yes?
A lot of genetically modified animals, like I've seen the documentary, and the pigs did it, the very first pig, and a lot of them died. They had to try so many times. So if they do it in humans, so
Right. Okay. So that's a great question. All right. So this is why this big flurry of activity right now with this CRISPR is that there's it's not, so the idea is, can you actually go in surgically and make one change and not have any other impacts, right? And what you're talking about is, there's a book called Frankenstein's Cat, is that right? Which is very much about all these things. People want to clone their cat, so they can have, they love their cat, they want to have another one, right? And it turns out that if you do that, it gets all screwed up, right? And so what you're saying is you do this one genetic engineering thing and something else goes wrong. Right? So right now, that would indeed be the case. But they feel, and that's why the Chinese are not working with embryos that could become humans. So they're tweaking these systems so that there wouldn't be off-target effects, where they do that. But I'm yet to be convinced, but I guess that's why I say to you folks, when you're my age, I don't think that's going to be a problem.
apes,
What's that?
like we can try apes.
Oh, yeah. Well, and some people would be very opposed to you doing that, right? Yes.
So does ethics drive or hamper science in general? Like it sounds like it's a good thing, but it's a bittersweet relationship.
Yeah.
Yeah.
So?
I mean,
Yeah.
like the mosquitoes.
Yeah.
Couldn't you just if you just accidentally release it, what kind of?
Yeah. Well, so okay, I'll get to your point in a minute. But that's a really important point. So right now, the Gates Foundation has to spend millions of dollars doing this, right? So what happens is these technologies, and I'm saying this is the beginning. What happens when they become easier? This technology, I have to say, that experiment that was in Science on the fruit flies, that was done by one graduate student. And he wasn't even aiming at this big thing. He was just trying to make these experiments easier. He had to count flies in the lab, and if you have a recessive trait, it's hard to do it. So he was just trying to convert heterozygous into homozygous so that he wouldn't have to count so many flies. So if that becomes that easy, that means if I don't like starlings, can I just happen to release them? Yeah. So these are difficult questions, and you're going to have to deal with it. I probably won't. Yes.
When you was talking about his question, were you talking about like the sterility of like the animals, like if they change one thing, then it'd be like basically like sterile so they can't reproduce? Or?
Well, it's more that, okay, so you have the possibility that somebody's going to have BRCA genes that potentially cause breast cancer, right? So is there a way of getting rid of that bracket gene? So that would be great. But let's say you try to do that, but at the same time you're getting rid of that, you cause a cut in another piece of DNA, and the baby has another medical problem. So you've cured one thing, caused something else. How do you ensure that that doesn't happen? And this is what happened with gene therapy. People were very careful about the first gene therapy things were injecting the virus, and this one person died, I guess, and that shut down the whole field. So you have to be very careful about what you do.
But wouldn't that also, like, if they use stem cells, like, you know, they grew or harvested the good stem cells, they can put in for the bad ones and put in for the ones that they took out?
Wait, try that again?
Like for stem cells, like you say, like, if you like take out that one gene, can't you put something else in its place to?
Yeah, well, that's the whole idea, is that you would indeed do that, but you have to be sure that that's all you do. But if something else happens as a result of that, so this cutting is supposed to be very specific. But right now, once in a while, it'll cut someplace where it's not supposed to. So one thing that could be done is you could, of course, check on the embryos to make sure nothing else is happening. But all I tell you is just keep reading the newspaper. You'll be seeing a lot of this stuff coming up because people are going to really be wrestling with this. So, wait, let me give you a chance, and I'll come back.
So just kind of coming up with I'm a chemist, organic chemist, and I've read a lot about this aquaboning technology.
Yeah.
And when the FDA has no chemical reason not to release this, which is really unusual because they released a drug that has no efficacy just recently by popular opinion. Bad science. I read all their findings and it was really, really bad. It was horrible. Aquabounding did 5, 6, 7, 100 times much more research in exactly those being safe. They're all female, they're sterile, they can't get released into the wild. They don't get allowed, but a bunch of dingbats who can't conduct correct studies. Sorry, I've done medical drug testing. It's a rotten world, but they did bad science.
Right.
And just by popular opinion, forced the FDA to do something. It makes no sense. The science.
Right. Well, all I would say is that if you get into the circles in Washington, D.C., and you ask what happened with the aqua bounty stuff, most people are very quiet. Then you realize that basically the Obama administration is not ready because of public opinion. So, how many of you here would like to eat genetically engineered salmon? Hey, all right, man, Aqua Bounty would love you guys. How many of you would not want to eat genetically engineered salmon? Oh, twice you're voting. Yeah. Gross, is that the word you use?
Yeah, that's the word that came up in class last week when we started talking about biotechnology.
People call it the ick factor.
There's no chemical reason. It's identical to the wild type.
So why don't the Mexicans want genetically engineered corn? They would have less capitalism. Right, so it's not, but this is this issue, right? If we try, and this is, I'm immersed in this all the time lately, because if a lot of scientists feel like, wait, if I just explain it to people, if people just understood more, they would understand this, and it'd be okay. But there's a human factor here that doesn't work that way, and actually, people have done these kind of studies with global climate change, and they look at people in terms of what their general ideologies are, whether if they're Republicans or they're Democrats. And then they look at those Republicans and Democrats that know less and know more about global climate change. And the difference between them, as they know more, gets bigger, not smaller. And this is true in a lot of these areas because you accept information that fits your preconceived notion. So they've done these great studies. There's a guy at Yale, Dan Kahn, who gets a bunch of people in a room, and they have the same person, presenting data, right, and explaining something. And if that person is explaining things the way that you want, you think of that person as, because you see the credentials, you decide those credentials are great. If they're presenting data that's not what you want, you find things in their credentials, and you say, that person's not that good. Right? So people do this all the time. So I guess that's why this is what our center is all about, is dealing with those kinds of issues. And what is it? So if you're going to deal with that golden rice, you need to understand the culture that it's going into. And it could be that you have to figure out what culture would it fit. So there's an interesting thing going on in Uganda where they're developing their own bananas with higher beta carotene. So what happens if, like if it belongs to Uganda as opposed, so we worked in Peru, there was one administration that was coming in that was pro-GMO, but the people who won were against GMO, and basically it says, look, this is coming from outside. We'll do it ourselves. We're going to take a 10-year moratorium, and it's going to be ours. So there's so much of that. I think if the Mexicans invented this, it would be different. So science is not done in a vacuum. So that's a major thing to recognize. You could have all the logic you think on your side, and also you could be wrong. So this is the other piece, right? It's that all of us scientists, entomologists have a little humility because we brought you DDT, right? And we thought it was great. Right? We're going to get rid of malaria. We're going to get rid of lice.
It's spreading into Florida all the time.
Right. So things change, and we thought we had estrogen therapy, right, for people going through menopause, and it would be wonderful. So what about genetics? We think this is going to be great. So I will just tell you one story. So we were doing all this testing on the corn that has BT in it, right? So we know that this bacteria kills caterpillars. But where do you find that bacteria? You find it in the soil. Well, caterpillars don't eat in the soil, they eat on leaves. So why does this bacteria produce this crystal of toxin that's 10% of its protein in the soil? Well, people have found that it actually affects some nematodes and it may affect some bacteria. 10 years ago, we thought we had a digestive system that had acids in it and all this other kind of stuff. We also know that it has huge numbers of different bacteria. Right? And we know that when you take the antibiotics, you mess that up. What do we know about eating corn that has BT in it? Would that mess up your gut bacteria? Well, recently people have done a lot of studies and found that it doesn't. But 10 years ago, we wouldn't even know to ask that question. So we have to, this has to be a science of humility in the sense that you, instead of saying, don't be stupid, listen to me, right? And saying, based on the science we have today, fine. But I can't promise you everything. So I think that's, yes?
If we conduct the research in a third world country and the government agrees to build the US, it will decline.
Okay, so the question is, if a government in a poor country wants genetic engineering of some kind, would the US sponsor it?
Yes.
Yeah. So that's a curious kind of question. So Monsanto and other high-tech companies want markets in those countries. So if a country wanted it, certainly we sort of arrange for those crops to be grown there. But in terms of public research, it's a whole other question. And there is units in the USDA that do research to help in that way. So at NC State, we have a sweet potato breeding program, which is doing just exactly what you're talking about, is breeding sweet potatoes for better qualities for developing countries. And if a developing country wants that technology, it's available. So tomatoes, potatoes, there are certain things that aren't profitable for a big company, but public research can do it. And there's a lot of question about whether we're decreasing the amount of funding for those things. Yeah.
First thing I want to say is you exactly stole my question that I was going to ask about the milkweed, the caterpillars and all that, so that's fine. You already answered that for me. Second thing is not necessarily actual, I guess, a question itself, but kind of walk me through the process of looking at genetic modification. But you also have to understand, obviously, that you're using on very, very large swaths of land where there are lots and lots of different other organisms, everything from caterpillars to microscopic organisms. Can you walk through kind of the process of what you guys would say is, okay, we're introducing these organisms, we're introducing these GMOs. What is the methodology that you're using and understanding how that effect is going to happen on the other surrounding habitats? You see what I mean?
Right. So actually, so I've been reviewing a lot of the research that's been done on, and it keeps being more and more research. You can get really bogged down reading it all, right? And so some of that research is done for regulatory compliance. So a company needs to do certain things to come to the EPA or come to the USDA or the FDA. So I could spend an hour talking about this. We have what's called the coordinated framework in the US, where for food safety it goes to FDA. For environmental effects, like how it's going to affect the surrounding environment, mostly to USDA, unless it's pesticidal. So if it's BT, it goes to EPA because it's a pesticide. And the people who have to be concerned about monarch butterflies would be the EPA and the USDA.
Right.
So the interesting thing, though, to get at your point, right? So if you have a BT crop, there's been a lot of work on the monarch butterflies. But if you create a crop that is resistant to a herbicide, glyphosate, Roundup, right? That goes through USDA. EPA has nothing to say about that crop because it's not a pesticide itself. What EPA has control over is glyphosate. So if you're going to have now a new use for glyphosate, because it used to be sprayed only on weeds before the crop came up because it could kill the crop. Now it's being more broadly used. EPA has regulation on that. So they then look at the possibility that by using Roundup more broadly, you're going to kill off the milkweed plants. And in the beginning, they didn't really do much in terms of that regulation. But now there's more push because the estimates are that we have, in Iowa, they did a detailed study. The estimates are that now there's 20% the amount of milkweed available than there used to be. Right. And you see that in Mexico, the numbers in the overwintering habitats are going down. And the question is that what's causing it?
Right.
There are just 6 new studies just came out, and these are all just academic studies that were just published all together that looked at the biology of the monarch butterflies in the US and Canada and can't find that actually decrease in milkweed is having that impact. But they're looking at it. But so to say, some of the work is done regulatory-wise, but we talk about this thing called governance, and governance is broader than that, and that's where the public has input. It doesn't have to be research done by the regulatory authorities.
Yeah, and just a quick follow-up on that is that I actually used to work at Home Depot when I was in the garden section, and I used to happen to be able to sell any of the Roundup with glyphosate, and it's just a short story. I had somebody, a neighbor, there was a customer that came in and sprayed it all along her fence, her chain link fence, and it was this person's neighbor that did that, and there was a hill there, and they sprayed it, and they were looking for like a selective herbicide, but this woman just set up spraying Roundup. It rained like 4 or 5 hours later, and all the Roundup washed down the hill and killed all of her grass. It's designed, it's engineered to be, to not degrade, to be as most survivable as possible.
Yeah, well, this is a big question right now, actually, because if you have that genetically engineered corn that survives it, but your neighbor doesn't have it, who's responsible?
Right.
So we just had a big meeting at NC State to deal with this.
It's 3 o'clock.
Yeah.
I know